Hi everyone, I am Sara El-Metwally, Assistant Professor in Computer Science Department, Mansoura University, Egypt. Today, I would like to share with you some thoughts from my recently published paper in current Bioinformatics journal. Our motivation to this paper is there is a plethora of sequence assemblers that has been developed to overcome the limitation of current sequencing technology. Each assembler tries to find the perfect answer for connecting a set of sequencing reads, and no tools guarantee to find the perfect answer since genome assembly puzzle is not a trivial task. These assembly results should be evaluated carefully before going a step further for any downstream data analysis. When evaluating an assembly results, four important questions will come to your mind. First, what percent of genomic composition elements are assembled? Second, how many contamination sequences are presented in your final assembly results? Third, how many errors are detected in the final assembly results? And finally, how many context scaffolds are resulted and their corresponding length? These four important questions will shape four important assembly evaluation metrics, which are accuracy, completeness, contamination, and contiguity. In this paper, we discuss the assembly evaluation process as a core stage in any sequence assembly pipeline. Also, we presented a roadmap that's followed by most assembly evaluation tools to assess different assembly evaluation metrics. Further, we highlighted different challenges that currently face the field of genome assembly and assembly evaluation tools. We summarized technical and practical details for different assembly evaluation tools in order to help developers and researchers to choose the best tool according to their working scenarios. I hope you all enjoy reading our paper in current Bioinformatics journal. Thank you.